In 5.2, we're looking at active transport. 5.1 was about passive transport, which didn't require energy, and we were going from areas of high concentration to low concentration, which is a natural flow. In active transport, we are not doing that. Active transport, the cell, it's going to require energy because here we're going to have the molecules go from areas of low to high, which is not what they naturally want to do. One way that we can cause this to happen is with a pump. And so we will use a pump to transport ions or particles or molecules across the cell membrane. In this case, we're going to be transporting sodium and potassium. And so the cell needs a certain amount of sodium and potassium ions. And so it's going to need high amounts of sodium outside the cell and low amounts of potassium inside of the cell. So it's going to use this pump to make that happen.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
So here is another picture of that. So again, that pump is going to move the sodium and potassium across that cell membrane. Another form of moving molecules and particles across the cell is by a process called endocytosis. So what's going to happen is those particles are going to come near that cell membrane, and the cell membrane is going to actually fold in and make a little pocket, and several of those particles are going to go inside, and then that cell membrane is going to move together and basically pinch off and form a vesicle to transport those macromolecules or large particles into the cell. So it's endocytosis is taking things into the cell. So again, the particles move close to the cell membrane, it folds in, forms a pouch, and makes that vesicle or a small sac that takes items into the cell. There are 2 main types of endocytosis, and I typically think of these as the cell eating or drinking. So penocytosis is where it's going to take in fluids, so that's like drinking for the cell. And phagocytosis is going to be solid particles. And so this is kind of like the cell eating, taking those particles in. Phagocytes often are going to take in things like bacteria and digest them, or viruses, and to keep them from overcoming the cell in a form of, combating, you could say. Also, lysosomes are going to fuse to those and then destroy those bacteria and viruses. And so in the next slide, you're going to see an example of this happening. So this main blob is the cell, and this little guy bouncing around is some type of bacteria that's trying to get into the cell. So you have this phagocyte that's going to try to engulf the bacteria and destroy it. So ultimately, he will be destroyed by the phagocyte. That was super dramatic, I know. The next type of transport we're going to look at is exocytosis, or exiting the cell. So just like endocytosis brought things into the cell, exocytosis is going to be a process where things are taken out. So endocytosis, the cell membrane created a pouch and brought things in. And now those vesicles are going to fuse to the membrane and then become part of the membrane and release anything, toxins, waste that might need to go out of the cell.